All right, good afternoon, everyone. It's officially 2 o'clock Central Time, so we will start today's weekly webinar. Happy November 2nd. Thank you for joining us today. Housekeeping notes, we are recording so that this presentation will be available for rewatching or for sharing. If you need closed captioning, the button at the bottom of your screen is there, and you can select it to show captions. Use the chat function if you have questions or comments while we have our wonderful presenter, and I'll monitor that. And I think those are all the announcements I have for right now. And before we end, I will talk to everyone about what next week's topic is, which is very exciting. But I'll keep you in suspense a little bit. Some of you might already know, but that just makes it more dramatic to make you wait till the end. In the meantime, please help me welcome our presenter for today, Dr. Tiffany Schwasinger Smith. And she is a double doctor. I just made that up right now. She has both a PhD and an MD. And Dr. Tiffany's PhD is in genetics, genomics, and bioinformatics from the University of Nebraska Lincoln, and she earned that in 2010. Dr. Tiffany got her medical degree at, of course, KU School of Medicine in 2014 and went on to complete her internal medicine residency at the University of Kansas School of Medicine in Wichita, where she is now an associate professor of medicine. And as you will find out shortly, that is the director of the Center for Clinical Research at the KU School of Medicine in Wichita. She's actively engaged in clinical trials research in the areas of psychiatry, neurology, infectious disease, and general internal medicine. She has been the principal investigator on more than 25 clinical trials with pharmaceutical companies, National Institute of Health, and private foundations. So you can tell that she is a good person to have on our team. Thank you, Dr. Tiffany. I'm going to turn it over to you. We're excited to hear about your wonderful work.
Well, thank you so much for that incredible introduction. And thank you all for having me today to talk about the research that we're doing, not only in clinical trials, but also the work that we're doing here in Wichita. I do want to have my financial disclosures. So I do clinical trials research. So I work with a number of pharmaceutical industries, the National Institute of Health, as well as private foundations. All of these contracts are with the university and not me personally, but I just want to make sure that I'm upfront in disclosing that. So what we're going to talk about today is kind of do a brief history of human subjects research. So how did clinical trials really get started? How did we start doing human-based research? And what are the things that we have to think about, especially with regards to ethics, when we start thinking about human subjects research and making sure that we're protecting people that are participating in clinical trials? We'll also kind of talk about the clinical trial process because it can be a little bit confusing when you hear about it. I know there's been a lot of mention in it of clinical trials and kind of the way that they work since we've started the pandemic. So I want to clear some of that up for you. And then, of course, talk about the great research that we're doing here in Wichita in collaboration with Kansas City and the ADRC. We're so excited to be partners in research. So when we look at the history of clinical trials, the first clinical trial that was ever recorded on record occurred in 500 BC. And so what was happening at that time is there were a lot of kind of wars, warriors, conquering of land and things going on at that time. And at the time, the diet predominantly was really based in wine as well as meat, as you could well imagine, because that was what was available and well sourced at that time. But at the time, a lot of the emperors and a lot of the leaders got together and started seeing what's going to make our warriors stronger, because we want to be stronger, better, faster. And so what they did is they actually did a 10-day intervention where they actually tried doing the standard diet, which was more wine and meat based, versus doing a more plant and water based diet. And to no surprise to anybody, people that were eating naturally actually had increased strength and better performance over time. So this was really the first human study that guided public health recommendations. And it's incredible to see something so simple could make such a difference. And it's still the things that we're using today when we start talking about dietary recommendations, eating closer to nature, getting plenty of water in, and kind of avoiding some of those higher fatty foods as we go through them. Probably the clinical trial that's the most referenced and the one we actually celebrate every year in May is the clinical trial that was conducted by James Lind. And this was really looking at scurvy. So what was happening is as sailors were going out into the open ocean, they were developing this disease. And you can actually see the picture here on my screen. where they'd have connective tissue and skin disorders. So they'd start having lesions and they'd start getting very, very sick on these ships. And at the time, they really couldn't figure out what was causing this. So James Lind, who was a physician, conducted this first clinical trial. And what he did is he took 12 different sailors who were on a sailing vessel, gave them a common diet. So they had a standard base of water, vegetables, meats as well. And then he started supplementing a few to see if any of these interventions would actually help. So 2 of the patients got a quart of cider daily. 2 of them got vitriol, which is basically kind of a fat-based mixture from animals. 2 of them got 2 spoonfuls of vinegar 3 times a day. Boy, would I not like to be in that group, I just have to say. 2 of them were supplemented with seawater. 2 received oranges and a lemon, and 2 received a honey-based medicinal compound. And what he actually found is what we now know about scurvy, which is caused by a vitamin C deficiency. And not having enough vitamin C, like you would not find when you're not on land, can cause these skin-based lesions and cause people to get very ill and die. So being able to supplement them with citrus actually prevented this disease from developing. So from there on, sometimes you'll hear the term limeys when it refers to sailors. It's actually because they found that limes were cheaper and had longer shelf life. So every ship that would leave, they would send them out with a bunch of limes to be able to supplement that and prevent scurvy. Now, we often talk in clinical trials a lot about placebo, because the way that we know a medication is going to work or not is we have to compare it to a placebo, or what's known as a sugar pill. And the first placebo-based clinical trials occurred with Austin Flint in 1863, where he was looking at patients with rheumatism, so rheumatoid arthritis. And at the time, what was the standard of treatment was actually ground up adrenal glands from animals, and that had cortisol in it, which is essentially prednisone or cortisone, which we use to treat a lot of rheumatic diseases. And he compared that against his control, which was an herbal extract. And what he found is that the people that were getting the steroid based or the cortisol actually did much better on it. So this is the first time that they actually used a placebo control in clinical trials. We move a little bit forward to looking at the double-blind control. So it's really important when we conduct research that we don't kind of insert our own opinions on it as the investigators or as the participants in the study. So what this was the first time where really the investigators didn't know and the participants didn't know. So this was conducted a study in the United Kingdom from 1943 to 1944. And what they were looking at was the spread of the common cold. So what they saw is people that were in factories or in close working conditions, the common cold, as we know and we see today, really spreads very rapidly through these populations. So they wanted to try something to see if they could prevent that disease spread. And so what they used was Ptolan, which is actually a penicillin-based extract, to treat it. And they took over 1,000 British office and factory workers, and they did a non-randomizing alternating treatment. So one person would walk up, they would get the Ptolan. The next person would walk up, they would get the placebo. Person after that gets the Ptolan. So there really wasn't anything other than just distributing each other person as they wanted to. Now, as we know, this was a negative trial because common cold is due to a virus. So antibiotics aren't going to help here. But it was really the first time where the investigator didn't know what the person was getting, and neither did the person. So that's really important in clinical trials, especially when we're trying to determine if medications are safe and effective. The randomized control trial, which is what a lot of people talk about, is really where we take it one step further. And so instead of giving out the medication one by one, it goes into a central area where they basically do like a database, and it randomly assigns treatment to each one, whether it's placebo or it's the active control. And the first study that actually implemented this type of design was when they were using streptomycin to treat pulmonary tuberculosis. And the reason that they could do this ethically, and we'll get into the ethics and concerns about ethics in research in a little bit, is that there was a limited amount of streptomycin, which is what was the standard of treatment during the time of the study in 1947. And so having that limited availability of the known treatment available allowed them to do the placebo control, where they could actually determine if the medication was working for treating the tuberculosis. They had a systematic enrollment criteria and data collection, and they actually used independent physicians to do objective measures looking at the x-ray. So as you can see on the x-ray image here, this is a classic x-ray image of a patient with tuberculosis. So you can see kind of that patchiness in the upper portion of the lung. That is a classic finding there. So they really took those independent physicians to review those x-rays to determine did they have tuberculosis or not, and was there improvement of symptoms or not based on that. All right, so the big thing when we start talking about clinical trials and human-based research is there have been some bad things that have happened over time, and we really need to pay attention to this to prevent it from happening again. So I think it's really, really important that we talk about where human research went wrong and the protections we've now put in place to make sure that we're taking good care of anybody who's participating in clinical trials or human-based research. The most commonly cited one, of course, is what happened during the Holocaust. So unfortunately, at that time, in the name of science, physicians did horrible atrocities to people, trying to understand the limitations of the human body. Most people do know that the Nuremberg trials occurred in 1945 to 1949. It was an international military tribunal, series of 13 trials, where people that were involved with the Holocaust and the concentration camps, they were charged with crimes against peace and humanity. But what I think is even more important to note is the separate doctors' trials that occurred from 1946 to 47. Here in these trials, they took 23 different physicians who had conducted research on participants without their consent and tried them for crimes against humanity. So I think it's really, really important to recognize the past and recognize where people have really crossed the line in research, and again, how we can help protect them. Another common example that people often reference when we start talking about ethics in research is the Tuskegee syphilis study. And this was actually conducted by the US Public Health Service from 1932 to 1972. And what happened is they took 600 low-income African-American males that were residing in Macon County, Alabama, and infected 399 of them with syphilis. And the goal behind the study was really to track disease progression. The problem with this is that penicillin, which is a known treatment for syphilis, became available in 1947, and it was not administered to the participants within the study. So unfortunately, 28 people died from syphilis, 100 participants died from related conditions, 40 spouses contracted the disease, and 19 children were born. Even further of where we went wrong are the Willowbrook hepatitis studies. So what this was a center for disabled children, either mentally or physically disabled, where parents could bring these children and they could get care. At the time during the Willowbrook experiments, hepatitis, there were some outbreaks there. And so when the parents brought their children there, they were told that, we're having a hepatitis outbreak, your child could get, sick and infected, but we're testing this new compound called gamma globulin, and we're hoping that it's going to be able to treat them. The problem with this is it wasn't just the natural disease spread that was occurring. They actually inoculated these kids with hepatitis, meaning they put it in their cereal, fed it to them, and infected them. So 700 children ages 3 to 10, 90% of those kids in the facility had hepatitis through these experiments. And this is really where we started talking about coercion, because these parents were not informed of what was happening to their children. They did not agree that these children needed to be knowingly infected with this virus and then tested on with this molecule that they were using for treatment. And this is also where we really started talking about risk and benefits of the research, because there was absolutely no benefit to these children participating in research. So now that we talked about the horrible things, and I'm sorry to kind of bring that downer, but I think it's always important to kind of reference things that have happened badly in research and really how we can move forward in a positive way and make sure that we're protecting people. So a lot of changes really went into place to protect human subjects that participate in the research. So this is a very busy slide, and I apologize for that, but this is the Nuremberg Code that went in 1947. And it's a lot of words, but what it really does is it clearly lays out informed consent. So people that are participating in clinical trials research or human subjects-based research need to know every single thing that will happen to them in the study. How many times I'm going to ask them a question about their depression? Exactly how many teaspoons of blood am I going to draw? And what am I going to do with that blood once I get it? How many times I'm going to give them a phone call? How many times I ask them to fill out a survey? And what are the risks and the benefits of participating in the study? Clearly laying everything out. And because we want to make sure anybody that's participating in research knows exactly everything that's going to happen to them, and they're okay with proceeding forward with it. This also really lays out kind of that risk-benefit ratio that we talked about. All research that we're doing should show added benefits and reduced risks. for participants in the study. And so this is really laying out that before it ever comes to human studies, it has to go through animal studies. So we have to be able to prove our concept in a model that is similar to humans in the way that it processes the metabolites or the different things that are occurring within the disease, and that the disease is similar to what we see in humans, and making sure that we have that data before we ever bring it into studies. Avoiding all unnecessary physical and mental suffering. So that has to be priority number one. We don't want to hurt people in trials. We want to make sure that we're answering the questions that we need to. Also making sure that there's adequate facilities and people are adequately trained. So it's not just someone who's never done research out there doing this, but it's people who have experience in research, who have medical degrees overseeing it in case anything happens that we can intervene, people that know what they're doing and that the science is solid behind it. Also allowing participants to withdraw at any time. So that's really, really key when you do clinical trials research, is at any point in time, for any reason, people can say, I don't want to participate anymore. So it's really important that we protect that right for them. And if there's any signs that things are going wrong, that there is a danger, that there's an increased risk for participants, we stop the trial immediately. And I think that is so incredibly key. And we've seen that in clinical trials. So I always reference the vaccine trials because that's probably the most relevant for the COVID-19 virus that we're dealing with. A number of those trials actually went on pause because there was a safety event where someone had something happen. And we took a lot of time, independent monitors of physicians, going through that data, making sure that it was safe to proceed. So I think that's a really great feature and a great protection that we put in research, making sure that we're taking care of people. I went a step further with the Declaration of Helsinki. So in 1964, the World Medical Association put together international research guidelines. So these were no longer just what we're seeing within the US, but agreed upon across the world. And this is the basis for good clinical practice. So again, making sure that those benefits outweigh the risk, making sure that people have training, making sure that we have the data behind it to say that this is going to be beneficial in people. Again, establishing that lab or animal-based research initially. All the protocols that we write are reviewed by independent committees. So it's not just me saying, hey, I'm doing great research, trust me on this. It's a lot of other physicians looking at it and saying, this makes sense, this is logical, this is going to be safe for people moving forward. And if we find anything that shows that it's unsafe, we have the ability to stop it. Again, showing that it's by qualified individuals, so making sure that you have the experience, the expertise, the equipment, the staff, the time, everything you need to conduct this safely. And risk, again, should not exceed the benefits. We took it one step further in 1976 with the Belmont Report, and this is where we really brought in medical ethics as well. So when we talk about medical ethics, there's a few that we always talk about. So the first is respect for the person. So individual respect. Everybody gets a right to choose whether they want to participate or not. And this becomes really important, especially if people have problems with cognition or aren't able to understand all the things that we're talking about, making sure that we have what we call an assent. So making sure that the participant agrees to participate, but if there's any question that they're not fully understanding it, that they have a caregiver or a representative to make sure that they understand everything that's going on and they agree for the participant being there. Beneficence, again, making sure that the benefit always outweighs the risk. And justice, making sure that everybody has access to clinical trials. This is something that we're really passionate about here in Wichita, is making sure that everyone, no matter where you live, including very rural Kansas, trust me, I grew up in Glenland, we're really looking at how do we engage people so that there aren't geographic or access barriers. So that's a big thing that we're focused on as we're conducting these trials. We also have what's called the common rule. And so this is really looking at making sure that the research is done correctly. So we have people that are kind of overlooking what we're doing in the clinical trials realm, making sure that we're following those protocols to the T, making sure that we're doing everything correctly. Looking at that informed consent, which is probably the most important document that we have in clinical trials. Again, so people know every single thing that we're going to ask them to do and every single sample we're going to take from them and what we're going to do with that information moving forward. It also defines IRB, which is kind of a separate committee that is out there to make sure that we're keeping people safe. So they look over everything that we're doing. If any safety events come up, we get in contact with them and the sponsor right away to report that. And then they make decisions as to whether or not the trial can continue or should stop. And then we also want to put in extra protection for vulnerable people. So pregnant women, prisoners or people that are in captivity, and children. making sure that we're taking care of those people and not taking advantage of them. Great. So let's briefly kind of go through kind of clinical trials. So when we talk about clinical trials, we want to talk about the research timeline. So the time from when it becomes an idea to actually getting out to market, on average takes about 12 years. So it's a very, very prolonged time and costs about $1 to $3 billion. So it is a substantial financial investment for a lot of these companies to be able to develop these drugs and devices. And it kind of goes through what we call the learn and confirm phase. So I took this from one of my partners, Dr. Sheldon Prescorn, but I think he sums it up very, very well. We start in discovery with an idea. What's a way that we can modify disease or treat a different pathway or, be able to alleviate symptoms? Going into that preclinical testing where we start doing the animal-based testing. And it's not just going to be in a single animal, it's going to be in multiple different animals that have similar pathways to humans. To again confirm that what we're doing makes sense and we're seeing that added benefit. And then to moving into the human studies, the phase 1, phase 2, phase 3, and then phase 4 post-marketing. So for drug discovery, this can really occur through a couple of different pathways. So new insights into a disease. So say we found a new receptor, we found a new molecule that kind of regulates how disease develops. A lot of it, though, actually comes from testing undeveloped molecular compounds. So most pharmaceutical companies just kind of have this library of stuff that they started working on for one reason or another, and they go back to that to see, potentially it didn't work for one indication, but it may work for something else. So they tend to go back to those tools. Also looking at new indications for approved medications. As a physician, a lot of times we use medication what we call off-label. The classic example of that is trazodone. It was developed initially for depression, but it causes a lot of sleepiness or sedation. So we often use it to treat insomnia, even though it's not FDA indicated for that. So again, looking for those new ways that old medications work. And then new technologies to alter disease. So this is classically, I like to talk about the Watchman device. So in your heart, you have a little area where sometimes blood can pool, and that blood can pool and form a clot and then go up to your brain and cause a stroke. In here, they can put a little device that prevents that blood from pooling, so that way people don't have to be on blood thinners lifelong. So that's a really great example of how we can use those new technologies to alter disease progression. When we talk about drug development, there's a couple things we have to think about. The first is pharmacokinetics, and basically that's a fancy way of saying what the body does to a drug. So we need to understand, where does it go in the body? So is it one that you have to put in as an injection in the muscle? Is it one that you can take as a pill? And then also metabolism. How does it break down? Does it go to the liver? Does it go to different areas of the body? Where is it distributed? So where are the receptors that it's going to affect? And then also looking at elimination because it can't stay in the body forever. And then also pharmacodynamics is what the drug does to the body. So looking at, can it reduce the enzymes that are processing it? Can it change the receptors? Can it change the messengers? Really kind of understanding how it works in the body and how it's processed. Now, when we talk about clinical trials, you'll often hear these different phases coming up. So I want to kind of break this down in a simpler process. Now, there's a lot of variability. So when we look at the new clinical trials, most of them are hybrid. So I've done phase 2, phase 3, phase 1, phase 3, all sorts of combinations. But basically, when we think about phase 1, those are going to be very, very small studies, 20 to 100 healthy people or people that have the condition. They're very short. They tend to be inpatient. And what they're going to do is they're going to administer a drug, and they're really looking for safety and dosage. So they're going to administer a drug. They're going to collect blood every 30 minutes to every hour and see, looking at that drug, when does it peak within the body where it has its full effect? When does it come down? Kind of looking at what doses are safe versus what side effects are we seeing at some of the lower doses or the higher doses, and how does it work? And when we look at these drugs, about 70% of those drugs move on to the next stage. So that's a pretty good number. But as we move through the clinical trials, you're going to see that number really drops to where we're actually getting drugs into market. So phase 2 is the bigger studies, several hundred volunteers, and these are normally people with the condition that you're trying to test. And it can last several months, up to 2 years, even up to 5 or 10 years, depending on the particular compound that they're testing. And here we're really looking at effectiveness. So how does it work in treating the condition that we're trying to treat? And also looking at side effects and safety. So if you've ever opened your prescriptions and you pull out the package insert and it says everything like, this drug can cause nausea, vomiting, diarrhea, and any number of other things. That's all information that we collect in the clinical trials because someone on the trial developed those symptoms and we have to determine is that potentially related or not to the medication. So in the phase 2, you can see there's a real drastic cut in the amount of medications that move on. So it really drops to about 33% of those drugs will move to the next phase. Phase 3 are the larger studies. So these are the ones that you heard with the COVID vaccine developments. These are the 300 to 3,000 to 30,000 people across the world that have the condition or we're trying to prevent the condition. And these are longer studies, about 1 to 4 years. Again, looking at effectiveness and monitoring if those side effects or adverse reactions. So again, really focused on safety. And again, of those 33% of the drugs that moved on, 25 to 30% of those moved to the next phase. And then we also look at phase 4, and those tend to be the post-marketing. So once it gets approved on the market, making sure it's still safe. If they see that there are increased side effects or risk to it, they will pull them off the market. So you'll hear about some of those drugs that get pulled from the market over time. Now I'm going to very briefly kind of go through this because I know we're getting tight on time. But all this goes to the FDA, which is the group in the US that really looks at these different medications and devices and makes sure that they're truly safe and effective for people and they should go out to marketing. So they look at all the data that comes through preclinical, animal trials, phase 1, phase 2, and they actually meet with the sponsors very, very regularly and tweak the clinical trials that we're doing. So if they see, for example, there's a cardiac effect, and we start monitoring cardiac enzymes and EKGs very closely. If they see that there's some inflammation going on, we start monitoring those labs. So they're really there to tailor it, looking at all the data and making sure that we're proceeding in a safe process. Once it's determined to be safe and effective, then it goes through a new drug application. And so this is where the FDA really digs down deep into the data. They review the risks, the informed consent, compliance with the trial, labeling, directions for use, safety reviews, drug abuse potential, what happens if you accidentally take too much of it, patient and provider information, as well as data that is happening in clinical trials outside of the US. And it takes them a lot of time to be able to go through this in depth. And a lot of times they go back to the sponsor and say, hey, we need more information. We need more data. We need another trial to occur. Now, fast track applications, I just very briefly want to talk about this because this came up a lot during COVID. It really reduces that review time, but I will tell you, it's just as safe as the other process. They look at it in complete depth, but these are things like serious or life-threatening diseases where we don't have a lot of other treatments. So especially when COVID was happening, this is where a lot of the research was going, is looking at this fast track indication, making sure that it's safe and effective, but kind of speeding up that timeline so that way we can take care of people sooner. And I do want to highlight, since we're on the MyAlliance, some amazing work that we've been doing here at KU Wichita for the past 10 years. So some of you may have heard of the new drug lecanemab or Leqembi that just received FDA approval as well as CMS Medicare coverage. So we've been working on that here in partnership with our participants here on the Wichita campus for the past 10 years. And I just want to highlight, this is the data that came out in the New England Journal of Medicine in January of this last year, did show significant improvements when we're starting to look at the CDR. So that's kind of a memory score that we use in research, that we do see that they're not having as much decline as what we see. When we look at the protein within the brain, which is, it is a binder of amyloid, we can actually see that it really reduces that here in the second graph. And then we also look at other cognitive scores, we can see that there is some improvement. So again, it's not curing the disease, but it's slowing down the progression of disease, which is really, really encouraging. It's so exciting to be a part of that and see that come to market. We do clinical trials. I'm going to do a little bit of shameless self-promotion for a second in all areas of medicine. And we are so excited to partner with our partners at the ADRC in Kansas City, doing Alzheimer's disease prevention, agitation. We're also looking at some more studies looking at mild cognitive impairment and how we can intervene earlier, biomarkers as well, looking at imaging to see if we can do early diagnosis. We also work with our partners in neurology and ALS, looking at treatment and biomarker studies. Of course, we're heavily involved in COVID, so vaccine studies, treatment, prevention, spread of the disease. We also do chronic cough, COPD, depression, inflammatory bowel disease, and so many more. So this is incredible that we're able to do this work in partnership with all of our colleagues here on the Wichita campus, as well as Kansas City and around the nation as we're doing clinical trials. And I always like to end with this slide. So at KU Center for Clinical Research, we are truly changing the face of medicine right here in Wichita. You don't have to go to Mayo Clinic. We're doing it here in Kansas. And rather than watching it happen, we're making it happen and helping our population be healthy. So, and with that, I will leave this QR code up. So if anybody's interested in learning more about clinical trials, learning more about the work that we're doing, you can just scan that QR code. We also have our phone number as well as our email here. So please, if you have any questions, do reach out. Thank you.
Excellent. Thank you so much, Dr. Tiffany. You make me be very proud to be associated with you and with all of your excellent work on clinical trials. For those in the audience who are watching who maybe are not KU staff, know that we have to do annual compliance and education training on what clinical trials are and how they work and all of the ethics. And I feel like, Dr. Tiffany, you've given me a good update on the training for this year. Thank you. And certainly you have given our audience lots of great information, and I think we'll all be quite grateful for the work that it takes to get a medication ready, and to feel safe and secure that it's going to be effective and not harmful and all of that stuff. I just want to take a moment to see if anyone has any questions or comments. You're welcome to unmute yourself if you do. I didn't see anything in the chat, but here's your chance to ask Dr. Tiffany something directly if you would like to. So we'll give anybody a moment to do that, and certainly give our thanks for your time today. Don't see anyone. Dr. Tiffany, I think you explained everything so clearly. Everybody feels like they are well equipped to move forward with this information. So thank you. Everyone take note of the email and the phone number on the screen, so that if you do think of some questions, that's the way to contact the folks in Wichita at the Center for Clinical Research. So keeping that in mind, I kept a lot of you in suspense. Next week's webinar, we are really excited, and I think a lot of you will be excited too. We are at long last going to be sharing our movie Why with you on next week's webinar. So Thursday, November 9th, mark your calendars. It will be in the Monday 11/13, I think. No, that can't be right. 11/6 issue of My Alliance. Monday, 11/6. We will officially be announcing that we are showing Why on the weekly webinar next Thursday. Some things to keep in mind. This will be in the newsletter. We will not be recording that session because we are not going to record the movie during the session. And the session will probably go for about an hour. The movie itself is 37 minutes. We'll watch it, and then we'll take some remaining time, maybe 25 minutes or so, for questions and comments. So that is the plan for next Thursday. So we won't record it, but we will show it. All you'll have to do is sign on to Zoom like you always do, and you'll be able to watch Why with us, and have a conversation with us about it. More details will be in the next My Alliance newsletter, so stay tuned. Tell your friends, spread the word. We're really excited, and we think you will find this movie meaningful and helpful. And Dr. Tiffany is nodding, so you know it's true. All right, so we are going to wrap up for the day. Thank you again, Dr. Tiffany. Thank all of you for being here. Everybody have a great weekend, and we'll see you next Thursday. Bye-bye.
Thank you so much, everybody. Take care.
Oh, Dr. Tiffany, make sure you look at the chat. You've got some kudos there.
Oh, yes. Thank you, everyone.
Yes. All righty.